The circulatory system is also called the cardiovascular system, where cardio refers to the heart and vascular refers to the blood vessels. So these are two key parts: the heart, which pumps blood, and the blood vessels, which carry blood to the body and return it back to the heart again. Ultimately, this is how nutrients like O2 or oxygen get pushed out to the organs and tissues that need it, and how waste like CO2 or carbon dioxide, which is the main byproduct of cellular respiration, gets removed. The heart itself is about the size of a person's fist, which makes sense. A bigger person has a bigger fist, and therefore a bigger heart. And it's shaped like a cone and sits slightly shifted over to the left side, in the mediastinum, which is in the middle of the chest cavity or thorax. It sits on top of the diaphragm, which is the main muscle that helps with breathing, behind the sternum or breastbone, in front of the vertebral column, squished in between two lungs, and is protected by the ribs. Now, if you look a little more closely, you can see that the heart sits inside a sac of fluid that has two walls, called the serous pericardium. The outer layer is called the parietal layer, and it gets stuck tightly to another layer called the fibrous pericardium, which is made of tough, dense connective tissue, which holds the heart in place and prevents it from overfilling with blood. The inner layer is called the visceral layer, and it gets stuck tightly to the heart itself, forming the epicardium, or the outer layer of the heart. The cells of the serous pericardium, both the parietal and visceral layer, secrete a protein-rich fluid that fills the space between those layers, and serves as a lubricant for the heart, allowing it to move around a bit with each heartbeat, without feeling too much friction. So, moving from the outside of the heart to the inside of the heart, after the epicardium, there's the myocardium, which is the muscular middle layer. This forms the bulk of the heart tissue, because those cardiac muscle cells contract and pump blood. In addition to cardiac muscle cells, there are crisscrossing connective tissue fibers, which are made of collagen that together form the fibrous cardiac skeleton, which helps support the muscle tissue. The myocardium also has dedicated blood vessels, called coronary vessels, which lay on the outside of the heart and then penetrate into the myocardium to bring blood to that layer, because it needs a lot of energy to pump blood. Finally, there's the innermost layer of the heart, called the endocardium, which is made of a relatively thin layer of endothelium, which is the same layer of cells that line the blood vessels. This endocardium lines the heart chambers as well as the heart valves. Alright, so looking at the right side of the heart, deoxygenated blood enters either through the top, through a blood vessel called the superior vena cava, or the bottom, through another blood vessel called the inferior vena cava, in the right atrium, where atrium means entryway. Both vena cavas are veins, which bring blood towards the heart. There's also a tiny third opening into the right atrium called the coronary sinus, which collects blood from coronary vessels returning from the myocardium. Now, all that blood then goes through the first of two atrioventricular valves that separate the atria from the ventricles. This one's called the tricuspid valve, and it allows blood into the right ventricle. The tricuspid valve has three little flaps or cusps, and each cusp looks kind of like a parachute because it has tiny little strings called chordae tendinei coming off of it that tether the cusp to a small muscle called the papillary muscle. When the heart contracts, that papillary muscle keeps the chordae tendinei taut, and both of these help to prevent regurgitation of blood back into the atrium, allowing it to only flow out the next valve. That being said, that contraction pumps the blood out the pulmonary valve, which just like the tricuspid valve has three cusps and also prevents blood from going backward. But unlike the tricuspid valve, the pulmonary valve doesn't have any of those chordae tendinei. Once it's past the pulmonary valve, the blood goes into the pulmonary arteries, which carry blood away from the heart to the left and right lung. Just remember that arteries start with an A, and also carry blood away from the heart. So that blood goes from the pulmonary artery into a pulmonary arteriole, which is a little bit smaller, and finally into a capillary, which is the smallest. In the lungs, the capillary lines up alongside a small sac of air called an alveolus, and when you have a lot of them, they're called alveoli. Up until now, that blood has been loaded up with carbon dioxide, which makes the blood look dark red, rather than blue, which is how it's usually drawn. And this is how we'll still draw it to stay consistent. Now, at this point in the journey, the carbon dioxide moves from the capillary to the alveolus, and oxygen moves from the alveolus to the capillary, giving the blood that nice bright red color. Now, in the blood, each red blood cell has millions of hemoglobin proteins, and each of those hemoglobins can bind to 4 oxygen molecules. So each red blood cell can carry millions of oxygen molecules when it's fully loaded. The oxygen-rich blood moves into a venule and then eventually into a pulmonary vein that dumps the blood into the left atrium. This trip from the right ventricle of the heart through the pulmonary artery to the lungs and back to the left atrium of the heart is called the pulmonary circulation. After entering the left atrium, the blood goes through the second atrioventricular valve, called the mitral valve, into the left ventricle. The mitral valve only has 2 cusps, or leaflets, one in front called the anterior leaflet, that's a little smaller, and one behind it called the posterior leaflet. Both of these have chordae tendineae coming off of them that tether the valve to the papillary muscles in the left ventricle. Similar to the right side of the heart, when it contracts, this prevents blood from going backwards. Finally, blood in the left ventricle gets pumped out through the aortic valve, which normally has 3 cusps, out to the aorta, the largest artery in the body. Just like in the lungs, the aorta branches into arterioles, which are smaller arteries, and finally into capillaries, which are the smallest, and at that point they're at the organs and tissues. In the organs, the red blood cells line up alongside tissue cells and drop off oxygen, as well as pick up carbon dioxide. Basically the reverse of what happens with the alveolus in the lung. Loaded up with carbon dioxide, the blood turns that dark red color again, shown as blue, and starts the return journey to the heart by going into small venules and then larger veins. Now, the lower half of the body drains into the inferior vena cava, and the upper half drains into the superior vena cava, both of which dump blood back into the right atrium. So this trip from the left ventricle of the heart to the body and back to the right atrium of the heart is called the systemic circulation. Relative to the pulmonary circulation, the systemic circulation has a lot more blood vessels, which means there's about a 5 times greater resistance to blood flow, which essentially means that it's a lot harder to pump blood through, even though it's the same amount of blood being pumped as the pulmonary side. Because of this difference, the left ventricle needs to be stronger, and so the muscular layer of the left ventricular wall, or its myocardium, is 3 times thicker than the right ventricle's myocardium. Okay, so let's talk a little bit about pumping. Each heartbeat sounds something like lub-dub, lub-dub, lub-dub. That first heart sound, lub, is called S1, and the noise comes from the tricuspid and mitral valves snapping shut when the left and right ventricles contract, which happens at about the same time. Right after the S1 sound, the aortic and pulmonic valve open up, allowing blood to get pushed out to the body, and this period of time is called systole. The second heart sound, dub, is called S2, and the noise comes from the aortic and pulmonic valves snapping shut to prevent blood from flowing backwards after it leaves the ventricles, effectively ending systole. Right after the S2 sound, the tricuspid and mitral valves open back up, allowing blood to fill up the ventricles again, and this period of time is called diastole. And that's it. Each heartbeat can be broken into systole and diastole. So systolic blood pressure is the pressure in the arteries when the ventricles are squeezing out blood under high pressure. And diastolic blood pressure is when the ventricles are filling up with more blood, so it's going to be slightly lower pressure. Alright, so if we simplify this guy a little bit, then the amount of blood that's pumped out by either ventricles over a period of time is called the cardiac output. And the rate at which veins return blood back to the atria is called venous return, which is pretty easy to remember, right? Since the circulatory system is a closed loop, cardiac output and venous return are equal. Okay, so let's use some numbers to make this a little more concrete. Let's say that about 70 milliliters is ejected per squeeze. With a heart rate of 70 beats per minute or 70 squeezes per minute, then 70 times 70 is 4,900 milliliters per minute, which in liters is 4.9 liters per minute, meaning that this heart's pumping about 4.9 liters per minute. Now, in an average adult, there's about 5 liters of blood total in the body, which, just to be clear, is different from the cardiac output we just found, which was amount pumped per minute. Now, taking this total volume, 10% of that, or about 0.5 liters, is in the pulmonary arteries, capillaries, and veins, which make up the pulmonic circulation. And 5%, about 0.25 liters, is in one of the four chambers of the heart itself. Another 15%, or 0.75 liters, is in the systemic arteries, traveling away from the heart. 5%, or 0.25 liters, is in the systemic capillaries, and the remaining 65%, or 3.25 liters, is in the systemic veins returning to the heart. Now, in terms of where all that systemic arterial blood goes, about 15% goes to the brain, 5% nourishes the heart itself, 25% goes to the kidneys, another 25% goes to the gastrointestinal organs, yet another 25% goes to the skeletal muscles, and the last 5% goes to the skin. These numbers can obviously change, for example during exercise, but this does give you a general sense of things. Alright, so that was the systemic arterial blood, but you'll notice that there's a lot more blood in the systemic veins. With that being said, arteries are generally lower volume while also being under much higher pressure. And in contrast, veins are usually high volume, low pressure vessels. And this explains why arteries and veins have different structures. For example, veins often have valves to help fight gravity and keep blood flowing in one direction back to the heart, whereas arteries don't need these valves because they're under higher pressure. If we take a closer look at blood vessels, they have three layers, also called tunics, or coverings, that surround the vessel lumen, which is the hollow part of the vessel that holds the blood. The innermost tunic is called the tunica intima, which includes the endothelial cells, which creates a slick surface that minimizes friction for blood moving through it. Next, there's the tunica media, or middle tunic, which is mostly made of smooth muscle cells and sheets of elastin protein. Both the cells of the tunica intima and tunica media generally get the nutrients they need from the blood in the lumen. Finally, there's the tunica externa, or outside tunic, which is made up of loosely woven fibers of collagen protein that protect and reinforce the blood vessel and anchor it in place. The tunica externa also has nerve fibers, lymphatic vessels, and, in the biggest vessels, the protein elastin. To make things really wild, some huge vessels have a tunica externa that's so thick that it needs its own blood supply. So there are tiny blood vessels called the vasa vasorum, which means vessels of the vessels, that creep along the tunica externa to bring nutrients to specifically that layer of the blood vessel wall. So the largest arteries closest to the heart, which are the aorta, its main branches, and the pulmonary arteries, have so much elastin in their tunica externa and tunica media that they're called elastic arteries. These guys are really stretchy, kind of like spandex. And this is what allows these arteries to keep their shape as well as absorb and even out the systolic and diastolic pressures. These arteries eventually branch into the arterioles, the smallest of the arteries. In the arterioles, the tunica media is bulky and can contract in response to hormones in the autonomic nervous system, which is called vasoconstriction, where the lumen gets a lot smaller, which decreases the blood to a capillary bed and ultimately the organ or tissue it supplies. Also, though, these vessels can relax or vasodilate, and the lumen's diameter increases, which allows more blood flow. This process can also help with temperature control or thermoregulation, because blood carries a lot of heat with it, and when it gets close to the surface of the skin, more of that heat gets lost. So with vasodilation of the arterioles, more heat gets lost, which therefore helps to lower body temperature. Likewise, if you wanted to raise body temperature, vasoconstriction reduces blood flow, which means less heat gets lost through the surface of the skin. Finally, the capillary walls are generally only one cell thick, just the tunica intima layer, with some larger vessels that also have a subendothelium basement membrane layer, which is a layer of protein just outside of the endothelial cells that provides extra support. In addition to allowing oxygen and carbon dioxide to flow back and forth, capillaries are also where nutrients like glucose can get delivered, and where fluid can move out of the blood vessel and into the interstitial space, which is the space between the blood vessels and cells. Water-soluble substances, like ions, cross the capillary wall through either water-filled spaces, called clefts, between the endothelial cells, or through large pores in the walls of fenestrated capillaries. Meanwhile, lipid or fat-soluble molecules like oxygen and carbon dioxide can dissolve and then diffuse across the endothelial cell membranes. On the other end of the capillary bed, there's a venule, and the arteriole and venule are usually directly connected by a vessel called the medarteriole. Alright, as a quick recap, the pulmonary circulation starts with the right ventricle, which delivers blood to the lungs, which then sends fresh oxygenated blood to the left atrium, at which point it enters the systemic circulation, starting with the left ventricle, which pumps blood to the body. And then deoxygenated blood comes back to the right atrium, and it all starts over again.
The cardiovascular system, also called the circulatory system, is the transportation system of the body. The major structures that make this possible are the heart, blood vessels, and blood. Simply put, the heart pumps the blood in order to move nutrients through the blood vessels to nourish and remove the metabolic wastes from the body. The heart has two major circuits within the circulatory pathway which work together in a closed circulatory system. These two pathways are the pulmonary pathway and the systemic pathway. In the human organism, the right side of the heart pushes the blood through the pulmonary circuit so that it can be oxygenated in the lungs. The left side of the heart pumps oxygenated blood to the entire body. The blood is then returned to the heart via the systemic pathway, where the cycle begins again through the pulmonary pathway. It can be said that the heart is actually two pumps divided by a septum or wall. The heart is a muscular organ with 4 hollow chambers. It consists of 2 atria, which are the upper chambers, and 2 ventricles, which are the lower chambers. The left and right sides of the heart are divided by the septum. The heart is also made up of 3 layers of tissues. The innermost layer is the endocardium, which is the thin, smooth membrane that lines the inside of the chambers of the heart and forms the surface of the valves, of which there are 4. Next is the myocardium, which is the middle layer. It's the muscular wall of the heart, or the heart muscle. And third, the pericardium. This is a tough, double-layered, fibrous sac which covers the heart. The word vessel has many meanings, but in anatomy and physiology, blood vessels are meant for carrying blood throughout the body. The blood vessels are similar to a two-way highway system. One lane, or direction, is known as the arteries, and the other is called the veins. Starting in the heart, the blood flows to the body through arterial blood vessels. The largest of these arteries is known as the aorta. These arteries provide the pathway for blood to travel to the body from the heart. Arteries are then further divided into arterioles and arterial capillaries. Arterioles are smaller blood vessels that branch out from larger arteries and lead to the arterial capillaries, which are the smallest blood vessels in the body. The arterial capillaries are connected to the venous capillaries. The arterial side of the capillaries is dropping off oxygen and nutrients to the body's cells. The venous side of the capillaries is picking up metabolic waste from the body's cells. Together they form a network where arteries and veins connect, completing the closed circulatory circuit. The blood flow from the venous capillaries will flow back from the body to the heart through the venous blood vessels. Similar to the arteries, only in reverse, the blood flows from the venous capillaries into a system of veins called the venules. These venules connect to larger veins which carry the blood back from the body to the heart via the largest veins known as the superior and inferior vena cava. Now the heart works alone, in that it has its own electrical system designed to work on its own, independent from the nervous system. If the nervous system is damaged and cannot send signals to the skeletal muscles or to other major organs such as the brain, the heart will not be affected because it has its own chemical/electrical system that will continue to pump the blood throughout the body. People with brain damage are placed on life support for breathing, not for pumping or contracting the heart. The only thing that would stop this mechanism is the lack of oxygen or a blockage or direct injury to the heart. The conductive pathway within the heart has 4 main stations through which it sends its electrical impulses in order for the heart to keep beating, which is roughly 100,000 times a day. The first conductive pathway is the SA node. The SA node, known as the pacemaker, is located in the right atria. It starts the spark and passes it on to the next relay station. The AV node is located on the back wall of the heart, between the right atria and the right ventricle. The spark is then passed into the next relay station, the bundle of His, located in the heart's septum, and lastly to the Purkinje fibers, which spread the electrical charge throughout the myocardium, which is the cardiac muscle, and causes the heart to contract, atrius first and then the ventricles. Here's a quick recap. The cardiovascular or circulatory system is an organ system whose basic purpose is to circulate blood to and from cells in the body in order to transport nutrients and remove waste. The heart pumps the blood to the body through the arteries, which branch off into smaller blood vessels called arterioles. The network of capillaries is where oxygen and other nutrients diffuse from the blood and into the cells. As oxygen and nutrients are diffused into the cells from the arterial capillaries, metabolic waste is diffused into the venous capillaries from the cells. The blood returns to the heart via the veins, where the cycle begins again. And remember that the heart has a beat of its own through the electrochemical circuit system and its 4 relay stations. To sum it all up, the heart pumps blood to the body through the arteries and returns from the body to the heart through the veins. Blood is a fluid or liquid known as plasma. Plasma is made up of 90% water, and this is why drinking a sufficient amount of healthy water is absolutely necessary. Blood contains many substances, but the 3 main substances we will discuss in this video are the 3 different types of blood cells, each having its own particular function. The first type of blood cell is called erythrocytes. These are known as the red blood cells. Leukocytes are known as the white blood cells, and thrombocytes are the platelets.
Let's say that I go to the doctor's office, and I hate when this happens, but I always, once in a while, have to have my blood drawn. And the reason I hate it is that I'm kind of a chicken when it comes to getting my blood drawn. I don't like needles. But of course, I do as I'm told, and I get my blood drawn. I just try to distract myself when the blood is actually filling that needle. And I usually look away, and before I know it, it's done, right? And so it's out of my mind, and I walk out of the office pretty happy because now I don't have to think about it anymore. But here's what I want to do now is kind of follow the path that this blood takes and think about what happens next after they draw the blood. So the first step is they usually put that blood into a tube, and usually that's done directly. Actually, these days, usually that tube is already kind of sitting and waiting, and it's collecting blood immediately. So this is my cap for my tube, and inside my tube I've got blood. This is my blood filling up this tube. And this is kind of a special tube, and the thing to know about this tube is that on the walls of the tube is a chemical that basically prevents the blood from clotting. You don't want the blood to clot because it's hard to do any sort of lab work on it, And so this tube is very special in that way. It won't actually clot. And so to make sure it's working properly, sometimes people kind of gently shake the tube up a little bit just to make sure that there's good mixing and so that the blood doesn't clot. Now from there, the blood goes over to the lab. So there's a machine in the lab that takes blood from me. This is my blood, but it also takes blood from other people. Let's say a few other patients in the hospital that day or in the clinic. And all of our blood is kind of labeled and put into this machine. And what the machine does is it spins. It basically spins really quickly. So all these tubes are attached. They don't fly away, but they basically spin as well. And if all these tubes are spinning, then what it creates is a force. Call it the centrifugal force. And so this process is called centrifugation. Let me write it out here. Centrifugation. And the machine is called a centrifuge. So it's basically going to spin really quickly, kind of, let's say, in one direction or the other. And as a result, what happens is that the blood starts separating out, and the heavy parts of blood kind of go to the tip of the tube, and the less dense part of blood actually rises towards the lid. So after you've centrifuged, let's say you've actually gone through this process, and you centrifuge the blood, now you have the same tube, but I'm going to show you kind of an after picture. So let's say this was before I actually spun the tube. And now I've got an after. This is my after picture. So after I spin the tube, what does it look like? Let me draw the tube. And the biggest kind of key difference here is that instead of having one similar looking kind of homogeneous liquid like we have before, now it actually starts looking really different. You've got 3 different layers. In fact, I'm going to draw all 3 layers for you. So this is the first layer, and this is the most impressive layer. The largest volume of our blood is going to be in this top layer. So remember, this is the least dense, right? It's not very dense, and that's why it stayed near the lid. And it's actually going to make up about 55% of our total volume. And we call it plasma. So if you've ever heard that word plasma, now you know what it means. And so if I was to take a drop of this stuff, let's say I took a little drop of this plasma, and I wanted to take a good hard look at what was in my drop. 90% of plasma is going to be nothing more than water. So that's interesting, right? Because the major part of blood is plasma, and the major part of plasma is water. So now you're seeing why it is that we always say, well, make sure you drink a lot of water, make sure you're hydrated, because a big part of your blood itself is water. In fact, that's true for the rest of your body as well, but I want to stress that it's true for blood as well. So that leaves the rest, right? We've got 90%, we have to get to 100. So 8%, what is 8% of this plasma made up of? It's protein. And let me give you some examples of this protein. So one would be, for example, albumin. And albumin, if you're not familiar with it, it's an important protein in your plasma that keeps the blood from, or the liquid from kind of leaking away out of the blood vessels. Another important protein would be antibody. And this, I'm sure you've heard of, but antibodies are basically involved in your immune system, making sure that you stay nice and healthy and don't get sick with infections. And another part of the protein, or another type of protein to kind of keep in mind would be fibrinogen. Fibrinogen. And this is one important protein involved in clotting. And there are actually many other clotting factors, we call them, as well. So I'm just going to put clotting factors here. So these are proteins, things like albumin, antibody, fibrinogen. These are all proteins. Now we've still got 2% to account for, and this is going to be things like hormones, for example, and that could be something like insulin. We've got electrolytes. That could be something like sodium. And we've got also nutrients. So nutrients, that could be something, for example, like glucose. So these things all make up your plasma. So a lot of the things that we kind of think about, talk about, are all in your plasma, including, vitamins and things like that. So now another layer we have right below the plasma is here in white. And if I was to kind of zoom in on it, it would be a very, very tiny part of blood, less than 1%. And this is actually white blood cells. This layer contains white blood cells and platelets. So these are cellular parts of our blood. And they make up a very tiny bit, but they're a very important part of our blood, of course. Below this layer, so now the most dense layer of blood would be the red blood cells. So this is this last bit, and this, just to make it add up, would be about 45%. About 45%. And these red blood cells, of course, contain within them hemoglobin, right? So sometimes it gets tricky because you forget, you think, oh, protein, so that must be plasma. Remember, red blood cells and white blood cells, within them, they have proteins as well. So just remember, they contain lots of protein as well within them. So for example, hemoglobin. So this is an example, right? Now, one word you may have heard of is serum. So what is serum exactly? Well, serum, this word, is very, very similar to plasma in terms of what it's made of. In fact, if I was to circle what is in serum, I would circle this bit. Basically, everything within my blue line would be circled. This is serum. And so the only thing I've left out of serum is fibrinogen and the clotting factors. So plasma and serum, you can just remember being very similar, and the exception is that the serum does not include the fibrinogen and clotting factors. Now, looking down at the red blood cells, what can we learn from that? Well, you may have heard this term hematocrit. And if this was my blood, if I actually had drawn my blood as I drew in this picture, and this was my blood, my hematocrit would have been 45%. And all that means is hematocrit equals volume taken up by red blood cells divided by the total volume. So if in this case my total volume is 100, my percent I already told you was 45%. So that's why I knew my hematocrit was 45%. It's just the percent taken up by your red blood cells. And that's an important percent to know because the red blood cells are the part of blood that are actually carrying oxygen around. Now, to kind of stress this point of hematocrit further and maybe even kind of introduce a couple of new words, let me draw out 3 little vials of blood. So let's say I have 3 vials here, 1, 2, 3. And these are going to be 3 different people, let's say. But they're all very similar, same age, same gender. Because hematocrit, what is normal, is actually going to change depending on whether you're talking about a certain age, a certain gender, even depending on where you live in terms of altitude. Because let's say you live at the top of a mountain, that is going to affect your hematocrit as well. So a lot of things affect hematocrit. But let's say we have 3 people kind of very similar in those ways. Now the first person, let's say I'm going to draw out their blood here. Their plasma, let's say, is taking up this much of their total volume. The second person, their plasma is taking up this much of their total volume. And the third person, their plasma is taking up, let's say, a lot of their total volume. Let's say all the way down to here. So you've spun all 3, and this is what you've gotten. Of course, all 3 still have white blood cells. Got to draw that in. And they have platelets. Of course, that's this tiny little layer, less than 1% we said. And the remainder then has to be red blood cells. So this is the red blood cell layer right here. This is the red blood cell layer. And it's really large here for this second individual. And this third individual, it's actually kind of on the smaller side. Not too much of the volume is taken up by red blood cells. So here, if I was to kind of go through and label these folks, I would say, well, this first person is what I would call normal. This second person has a lot of red blood cells. It's so predominant. This is a very high percentage, right? I know this is taking up a high amount, right, of the total volume. So this person has what I would call polycythemia. Polycythemia. It's just a medical word to say that the volume of red blood cells over the total volume is very high, or you could say their hematocrit is very high. And this person, this third person, has a very low amount of red blood cell volume relative to the total volume. This is actually pretty low. And so this person, I would say, has anemia. So if you've ever heard these terms, anemia or I'm anemic, sometimes people say, or even the word polycythemia, now you know it's just referring to what volume of their blood is taken up by red blood cells.
So, let's talk blood, shall we? For most purposes, blood comes in 8 different types, and it accounts for about 8% of your body weight. Blood is a type of connective tissue, which means it's made of living cells suspended in a non-living matrix, which in this case is the fluid ground substance called plasma. And of course, one of blood's main missions is to transport and distribute oxygen and nutrients and waste products and hormones around the body. But it also helps maintain and regulate body temperature, pH levels, and the volume of fluids in your body. Plus, it protects you from infection and from the loss of blood itself. So it has a whole system in place to prevent you from losing too much of it through a process known as hemostasis. So imagine you're slicing up a nice garlic cheese bagel one morning and you lacerate the distal phalanx of your pollex. In other words, you cut the tip of your thumb. And now you're bleeding all over your breakfast. At the very first sign of rupture, the blood vessel actually constricts itself to slow the flow of blood through it. Then little cell fragments called platelets gather at the site of the injury, creating a plug that dams the breach and keeps the blood from leaking further. Now, these free-floating platelets don't clump together during regular circulation, that would be terrible, but when the endothelial cells lining a blood vessel wall tear, the underlying collagen fibers are suddenly exposed, and they chemically react with the platelets, turning them all sticky and glue-like at the scene of the injury. But that platelet plug still isn't as strong as it could be. It needs reinforcement to complete the clotting process. This reinforcement comes in the form of fibrin threads, protein strands that join together to make a sort of mesh that traps the platelets and blood cells. Eventually, the threads actually pull the opposite sides of the wound together to close the vessel wall so the endothelial cells can be replaced. So you have A, B, or O. These different types all do the job equally well, they just sort of have a different flavor related to your immune system. All the cells in your body have a plasma membrane with specialized glycoprotein markers on them that act like name tags or labels, sort of like, this cell is property of Hank. These markers are your antigens, and your body's immune system is totally fine with your particular antigens, but if it detects antigens from someone else's cells, including viruses or bacteria, then it'll send out antibodies to bind to those markers, often to tag them for destruction by the immune system. Your red blood cells have specialized antigens on them called agglutinogens that activate antibodies that work by binding invading cells to each other, which causes coagulation, or the clumping of blood. Which agglutinogens you have on your erythrocytes defines your blood type, but they're classified in 2 different ways. In the most important blood classification, the kind people are most familiar with, there are only 2 kinds of agglutinogens, simply A and B, and your blood can either have one or both or neither of these molecules. So the name of your blood type refers to what kind of agglutinogens you have or don't have. A type has A antigens, B type has B, AB has both, and O has neither. So why do you need to know what type you are before you give or receive blood? Well, like I mentioned, if you have either of these antigens, your body will be fine with it because it doesn't produce any antibodies that label it for attack. So if you don't have a particular antigen on your blood cells, say the type B, then you do have antibodies that are going to label those B antigens for attack should they enter your space. So AB type folks are called universal recipients because they have both antigens and therefore no antibodies for either. So they can accept A or B or AB or O blood. Meanwhile, O type doesn't have A or B antigens, so those folks have antibodies for both. That means that they can only accept other O blood. And yet that lack of antigens means that type O blood can mix with other types of blood without getting attacked, which is why it's known as the universal donor. But just to complicate things a little bit more, you've got a whole other set of antigens with totally different protocol. These are your rhesus or Rh antigens, named after the species of monkey they were first identified in. Much like A and B, you either have the Rh antigens, in which case you're Rh positive, or you don't and are Rh negative. Most of the population is Rh positive, so they don't have the anti-Rh antibodies, which means they can accept either positive or negative blood, but negative types should stick to just the Rh negative blood. And since the presence of AB antigens is controlled by different genes than the Rh ones, we end up with 8 different blood types, 4 separate groups, each with 2 variations. Your blood is incredibly powerful stuff, and its power rests largely in your erythrocytes, or red blood cells. They're the most abundant type of cell in your blood, accounting for nearly 45% of its volume. Every time you take a breath, they pick up oxygen in your lungs and distribute it through your body, and then grab carbon dioxide and bring it back to the lungs where it can be exhaled. The main mission of erythrocytes is to keep your body fed with oxygen so your muscles can do their thing and your brain can continue to think and feel and boss around your various parts. But you don't want to mess around with your your red blood cells because erythrocytes are weird characters. They go places that other cells won't. They purge themselves of their most precious inner belongings, preferring instead to live as hollow shells. Because of the crushing demands of their job, they don't live very long, and just like with your blood pressure, too much of these good things can turn bad quickly. Your red blood cells are fairly simple and unassuming little cells. They've got a distinct biconcave shape, which just means that they're concave on both sides, making them look kinda like a breath mint. A tiny, bloody breath mint. And while they do have a plasma membrane, they don't have a nucleus and don't have most of the parts that other cells do. So they're basically just glorified protein-filled phospholipid bilayer sacs. But there's still another great example of that harmony between form and function. For one thing, that biconcave shape gives them a large surface area that's ideal for gas exchange. It also makes them flexible and able to change shape as they squeeze through tiny capillaries with diameters smaller than the cell itself. Of course, all that squeezing and twisting is hard on a cell's membrane, and that, combined with their general lack of organelles to help repair the membrane, means these cells don't live very long, surviving on average only 120 days. But they sure work hard while they're alive. And their work is mostly in gathering and transporting oxygen. They're able to do this because if you don't count their water content, red blood cells are 97% hemoglobin, a molecule that easily binds to and releases oxygen. It's like an oxygen sponge. Every hemoglobin molecule is really made of 8 different component molecules. 4 are a red pigment called heme, and 4 are a protein called, you guessed it, globin. Each globin is a globular polypeptide chain, hence its name, and proteins, you'll probably remember, like to bind to stuff. So each globin has its own personal ring-shaped heme molecule, and in the center of that heme is an iron atom, kind of like a cherry on top of a protein and pigment sundae. It's that iron in the center of the heme that makes our blood red. Each iron can bind with one whole oxygen molecule, and that oxygen really adds up. Since you have 4 iron atoms in every molecule of hemoglobin, and every red blood cell contains something like 250 million hemoglobin molecules, that means that each one of your tiny floppy red breath mints can grab about 1 billion molecules of oxygen. Exactly how they transfer oxygen and carbon dioxide from your tissue cells is something that we'll get into more when we talk about the respiratory system. But if you're wondering why all this hemoglobin can't simply skip the red blood cell rigmarole and just run around naked in your blood, it's because free-range hemoglobin would actually thicken the blood, making it so viscous that it would impede blood flow. So what does the brief but glorious 4-month life of an erythrocyte look like? Well, remember when I said that a red blood cell doesn't have a nucleus? And maybe you thought, hey, wait a second, how can it even be a cell without a nucleus, or DNA? First of all, nice catch. But actually, erythrocytes do start off with a nucleus and DNA. They just get rid of them because their entire purpose for existing is to schlep around hemoglobin and oxygen, and they want the extra room. The whole process of forming red blood cells, called hematopoiesis, happens in your red bone marrow, which is mostly made of reticular connective tissue that's snuggled up to special capillaries called blood sinusoids. In short, the process begins with a hemocytoblast, or a specialized stem cell, which soon differentiates into an early erythroblast. Then it starts making a whole bunch of ribosomes, the organelles that manufacture proteins. And in this case, the ribosomes start cooking up tons of hemoglobin as the cell transforms into a late-stage erythroblast. When it's got enough hemoglobin, it suddenly jettisons most of its organelles, which causes the cell walls to collapse a little, giving it its biconcave bloody breath mint shape. Now you're left with a reticulocyte, which is pretty much just an early erythrocyte that still has a little group of ribosomes left called a reticulum. So far, this whole journey has taken about 15 days. When the reticulocyte is finally just about bursting with hemoglobin, then it leaves the marrow and enters the bloodstream, and a couple of days later, when the last of the ribosomes have degraded, you've officially got yourself a mature red blood cell. And that cell travels around your body, doing its job for a few months before it gets old or damaged and needs to be replaced. Now, maintaining the balance between production and destruction of these cells is crucial. Too many will make the blood too viscous and difficult to pump, and too few leads to oxygen deprivation, or hypoxia. The process of maintaining the right levels of red blood cells is regulated by a special hormone called erythropoietin, or EPO. It's produced mostly in the kidneys, but also in the liver, and is constantly circulating in the blood. If you're anemic, or hiking at high altitude, or hemorrhaging blood, or experiencing anything else that creates a drop in your blood oxygen levels, certain cells in your kidney will take notice and take action. And they can do that because they traffic in a signaling molecule called hypoxia-inducible factor, which monitors your blood's levels of oxygen. The way that this works is pretty cool. These special kidney cells need oxygen in order to break down that signaling molecule. So if oxygen levels in the blood are low, they can't turn the signal off. This means that the signal keeps going, which triggers the release of more and more EPO, which stimulates your red bone bone marrow to pump out more red blood cells to carry around more oxygen. As oxygen levels in your blood increase, the signal is degraded and EPO production slows. So if you're generating around 2 million new blood cells every second, you also have to dispose of about the same number of dead ones to maintain the balance, right? When these cells get old, they turn rigid and their hemoglobins start to fall apart. As they get stiffer, they can end up getting stuck in capillaries in your brain or heart, which would not be good. Luckily, you have certain channels to corral these dying cells, especially around the spleen, which some anatomists call
the red blood cell graveyard.
So these tired old cells get trapped and then basically ambushed by big macrophage white blood cells in the spleen, liver, and bone marrow, which break them down and recycle their various components. The globin proteins are broken down into their basic components, amino acids, which go back into the blood to be used by other cells for making more proteins. Iron from the heme group is separated and either bound to proteins and stored in the liver, or put right back into a new hemoglobin molecule, and the heme gets turned into bilirubin, a yellowish pigment that goes to the liver where it's added to the bile that it secretes into the intestine and eventually leaves the body in your poop.